Hi students, this is A. Mandari Kumar Rajesh. In this video, we are going to discuss new chapter called Biotechnology. Let me give you the definition of biotechnology. Biotechnology deals with techniques of using live microorganisms or plant cells or animal cells or their components or enzymes from organisms to produce products and services useful to human beings. So the biotechnology can be defined as the techniques of using live microorganisms or plant cells or animal cells or their components or enzymes from organisms to produce products and services useful to human beings. So this is the first definition. Then I am going to give you another definition. It was given by European Federation of Biotechnology. So the definition was given by European Federation of Biotechnology. So according to EFB means European Federation of Biotechnology. Biotechnology is the integrated use of biochemistry, microbiology and engineering sciences in order to achieve the technological applications of the capabilities of the microorganisms. Then culture tissues or cells and parts thereof. So this is the definition which was given by European Federation of Biotechnology. Then the definition involves two common factors. The definition of biotechnology involves two common factors. The first factor tells us the technology has to use biological agents. Then the second factor tells us that technology must useful to the human beings. So the biotechnology has two important factors that tell us. The first factor tells us The technology has to use the biological agent. Then the second factor tells us that technology must useful to the human beings. Then the biotechnology, the term biotechnology was coined by Carl Erki. Carl Erki. He was an Hungarian engineer. He coined the term biotechnology in the year 1919. Then I am going to tell about the development of biotechnology. Let me tell about the development of biotechnology. Biotechnology has developed by leaps and bounds during the past century. That means it is a branch of science. It shows enormous growth. It shows enormous growth in limited period of time. In limited period of time. It shows vigorous growth in limited period of time. Then the development of biotechnology can be understood by two main heads, namely conventional biotechnology or traditional biotechnology Then the second heading is modern biotechnology. First, I am going to tell about the conventional or traditional biotechnology. It is a kitchen technology. It is otherwise called as a kitchen technology. It was developed by our ancestors. It is as old as human civilization. The technique is as old as human civilization. In that our ancestor used microorganism or bacteria for the production of dairy products like ghee, then cheese and curd, and also they used the microorganism for the production of idli, then dosa, then pizza and so on. For example, in our home, our mother used to pour some amount of the buttermilk, fermented buttermilk. In Tamil it is called oraimor. So in course of time, that milk, normal fresh milk, converted into curd. In overnight, the normal milk converted into curd due to the microorganism which are present in the fermented milk. So that microorganism converts lactose into lactic acid. And another example, in idli dough, our mother used to mix the idli dough, that is the rice flour dough and the black dough are mixed by our mother by her bare hands. So in her hands some probiotic microorganisms are present. So probiotic means the beneficial microorganism may be present, and she used to mix the dough by her bare hands. Then in the morning the dough looks somewhat effervescent. It shows the rise of dough, and it gives some fragrance, it gives some flavor. Due to the microorganism which are present in her hand, it makes the dough softer and it gives flavor to the dough, and it makes the dough so softer, and it's very tasty. So in olden days, our ancestors unknowingly using these techniques. With the advancement of science and technology in the 18th century, this kitchen techniques get validation. That means these techniques are accepted by the scientific world. Then I'm going to tell about modern biotechnology. Tell about modern biotechnology. Modern biotechnology contains two main techniques. They are genetic engineering. They are genetic engineering. Then chemical engineering. So genetic engineering means it is a technique includes the manipulation of the genetic material. It may be a DNA or RNA. Then the manipulated genetic material are inserted into the host cell, and thus it shows some changes in the phenotype of host. It shows some changes in the phenotype of host due to the alteration of genetic material, due to the manipulation of genetic material, and that technique is called as genetic engineering. While in the case of chemical engineering, they are maintaining the microbes in a sterile condition to avoid the contamination. Mainly, they are maintaining the microbes, the desired microbes, for the manufacturing of large quantities of biotechnological products like antibiotics, alcohol, vaccines, etc. So by using these techniques, we can produce biotechnological products like vaccine, antibodies, then alcohol. So these two techniques are very, very important in modern biotechnology. So you have to do these two techniques in the biotechnology. Then I am going to tell about the main focus of modern biotechnology. So please wait a minute. Let me teach you the main focus of biotechnology. Here I have written some of the process which can produce value added products. First, fermentation. Fermentation is an anaerobic process in which chemical substances are break down by microorganism, and it can produce acids, acetic acid or citric acid, and it can produce enzymes, alcohols, antibiotics, vitamins, etc. So fermentation is an anaerobic process in which chemical substances are break down by microorganism, and it can produce acids, enzymes, alcohols, and antibiotics. Then the second process, biomass. In this process, we are going to use a living organism, and that living organism it can produce in a large quantity by using bioreactor. We can culture that living organism in a large quantity by using bioreactor. By using that organism, we can produce single cell protein. So it is an alternative diet for that diabetic patients. So the diabetic patients can consume single cell protein. Then alcohol. By using biomass technique, we can produce alcohol and we can produce biofuel. Then the third process, enzyme as biosensors in processing industry. So enzyme can act as a biosensor because it can recognize and it can react with the target analyte. And it can give the signal, chemical signal. That chemical signal is converted into a physical signal by transducer. So that enzyme gives a chemical signal that is converted into a physical signal by transducer. So the physical signal means it makes changes in the physical appearance. By that appearance or by that signal we can understand the enzyme act as a biosensor. That enzyme react with that target analyte. By that signal we can understand that enzyme react with that target analyte. Then biofuel. So by using microorganisms or by using the waste product which are getting from that cow, or we can use some sugarcane molasses, even we can use water to make biofuel. So biofuel is a fuel which can produced by contemporary process, so we can use water, then sugarcane molasses to produce alcohol, then cow dung are used to produce methane gas, that is coal gas or biogas. Then microbial inoculants. So we can use microorganism for biofertilizers. So we can use microorganism as biofertilizers and nitrogen fixers. For example, Azolla anabina. Azolla is a pteridophyte, anabina is a cyanobacteria. So they make a symbiotic association. They can fix the atmospheric nitrogen and they can act as a biofertilizers. Otherwise, we can introduce some microorganisms which can act as a biofertilizers. Then plants and animal cell can produce secondary metabolites. They can produce secondary metabolites and monoclonal antibodies. Secondary metabolite means it is a product. It is not vital for their vital process. So it is a product produced by the organism. It is not important for their vital process, but it has a value added product. For example, for common cold, we used to make a decoction with herbs like Solanum trilobitum, Solanum surutans, Ocimum, that is Tuduvalai, then Kandangatri, then Tulasi. So by using these herbs we can make a decoction. Then by consuming that decoction we get relief from that common cold, because that decoction contains alkaloids. Alkaloids are nothing but the secondary metabolites. So the secondary metabolites contains value added products. So we can produce secondary metabolites from plants and animal cell. Then monoclonal antibodies. It is an important antibody. These monoclonal antibodies act as a substitute for immune cells. It can restore our immune system. It can attack on cancer cell. So the monoclonal antibodies are produced by hybridomo technology. So the monoclonal antibodies are very important antibodies to attack on cancer cell. So monoclonal antibodies are nothing but substitute cell for immune cells. Then recombinant DNA technology. By using recombinant DNA technology, we can produce enzymes, vaccines, growth hormones, antibiotics, and interferon. So interferons are very important proteins. Interferons are nothing but antiviral protein. So that antiviral protein can stop the activity of virus. So interferon is very important. Interferon means antiviral protein. Then by using recombinant technology we can produce enzymes, vaccines, and growth hormones. So by manipulating the gene we can produce value added product. For example, we can produce humalin, that is insulin, human insulin. So alter the genetic material, can insert it, that is the desired gene of the desired gene can inserted into the host cell, that is E. coli. E. coli is a bacteria. So that desired genes can inserted into the E. coli by vectors. By vectors, it can produce the desired value added product, because every 20 minutes E. coli get multiplied. So this plasmid contains the desired gene, and it can integrated with that E. coli DNA, and it can produce the value added product insulin. So every 20 minutes we can get a copy of insulin. We can get insulin every 20 minutes. Then process engineering techniques, that is process engineering techniques are tools of biotechnology, is used for effluent treatment and water recycling. That means that microorganisms are used in the techniques to treat effluent, then the microorganism can remove the contaminants from the effluents, because they can oxidize that contaminants, and they can remove the contaminants from effluents. And also in water recycling, we can use microorganism to remove the contaminants from water. So that's all about today's video. If you have any doubt, please clarify with me. Thank you very much for watching this video. Thank you.